So today I want to talk about estimation of inheritance components that with the models having fixed and random effects in them. And as I've told several people, this is a talk I usually do on a whiteboard, and I've turned it into a PowerPoint, and I don't want to go too fast. So if you feel like I'm going too quickly, please slow me down. And also feel free to jump in with questions as we go along. So if we use up the discussion time during the talk getting questions answered, that's probably an efficient use of time as opposed to going through it all and then going back. For you Enophiles out there, this is Châteauneuf-du-Pape in southern France. There's the Rhone River. This is looking from the hill where the chateau was, which the Germans thoughtfully blew in half in 1944 when they were retreating up the Rhone Valley. There's a lot of different ways to estimate variance components, and we talked about several of those yesterday. Analysis of variance is one way to partition phenotypic variance into genetic and environmental components. Another one that was discussed more thoroughly yesterday was regression, and as was noted yesterday, regression in fact was developed for estimating heritabilities. However, there are some limitations to these approaches, especially when you get large unbalanced data sets that have very deep pedigrees. And so you want to make use of all the information that you have in your data set, and if you've got multiple generations of family information, you want to be able to include that. And so with these large data sets that can be unbalanced and with deep pedigrees, we can use linear mixed models to estimate variance components. And the other thing that we can do with these models is that we can have other sorts of variables in there that were not in the examples used yesterday. So for example, if you have measured fixed effects in your populations, like different experimental groups or maybe different populations. In fact, Patrick was telling me yesterday with those snake data, those snakes were collected at a number of different sites within the broad study area, and they removed the effect of that variation before beginning the quantitative genetic analysis. Ideally, what you want to do is include that in your quantitative genetic analysis. So that would be a fixed effect that you want to put some of the appropriate amount of variation into. You also have, in addition to fixed effect, what are called random effects. And random effects are variables in which the units are randomly sampled. And in the linear mixed models that you use for quantitative genetic analyses, the most common type of random effect used are the individuals themselves. Because what you're saying is, okay, I have this population of individuals, I'm going to reach in and pull one out and use it for my analysis. But I really don't care about this individual. It's representative of all individuals, so I'm happy to put it back and pull out another one. So that's a classic example of a random effect, okay? So in these linear mixed models, I'm assuming most of you have dealt with them in some form or another before, but if you haven't, one thing that you need to think about before you begin is which of your variables are fixed effects and which are random effects, and sometimes it's not so easy to decide. For example, if you've measured the gender of the individuals in your population, should gender be a fixed effect or should it be a random effect? You could sort of argue either way. And so you need to there's some value judgments to be made and decisions to be made frequently about whether something is a fixed effect or a random effect. And there's not necessarily a clear-cut answer. Now, the type of linear mixed model that is frequently used in quantitative genetics is called the animal model. And it's called the animal model because it was developed by animal breeders, but it works equally well on plants and other types of organisms as well. So we're going to go through this so-called animal model a couple of times and talk about what these terms mean, and we'll have a little toy example and all that sort of stuff, because I want you to have some idea of how this works and not to be intimidated by it. So I want to go through first what this animal model is, and then we're going to go through an example of a very simple example of an animal model. Okay, so as we look, this is the so-called animal model. It's y equals x beta plus ZA plus E. So let's go through and talk about what each of these things are. And all of these things are going to be either vectors or matrices. And whether you know it or not, when you're using SAS or R or any of those things to do linear models, it's actually solving those equations using linear algebra. Okay, it's not using scalars, even though you tend to think of things in terms of scalars or just individual measures. It's really using matrix algebra to solve everything. So for example, Y is the vector of phenotypic measures. You go out into your population and you measure body mass or something like that. And you're probably used to thinking as body mass as a scalar. It's just one number. But in fact, the way that statistical programs think about it is it's a vector of measures, okay? So this looks scary, right? Oh my God, it's a vector of measures, but it's just a list of the data that you collected. It's no big deal, okay? We're just thinking about the entire list of those measures you collected instead of thinking about them one by one. So Y is the vector of phenotypic measures, and this phenotype whose variance you want to be able to partition into genetic and environmental components, or genetic and residual components, okay? Now, the terms in green are related to the fixed effects. Beta is a p/1 vector of fixed effect regression coefficients. So for example, if your fixed effect is location and you have two locations, then you're going to have your vector is going to have two elements to it, a beta 1 and a beta 2, one for location 1 and one for location 2. Okay, so these are regression coefficients. So this is your primary fixed effect. Now, one of the if you're not used to thinking about your data this way, the weird thing are these so-called design matrices. And what design matrices do is they relate either a fixed or random effect to the phenotype that you've measured. And I think this will make more sense when we look at an example. And it turns out these are very simple matrices. So X is an N by P, so N is the number of individuals in the population. P is the number of different fixed effects that you have. So it's an N by P design matrix relating Y to beta. And basically all it's telling you is whether or not, is telling you which of the betas, which of the fixed effect groupings that the individual on which you've collected that specific data point belongs to. And when we look at this in an example, all of a sudden the penny will drop and you'll go, oh my God, that's just trivial. And it is, but you need it in there to be able to solve the equation. So those are the fixed effects. The random effects I've shown in blue. And A is a q/1 vector of additive effects or breeding values. Those are the additive effects that give rise to the additive genetic variance. So in some ways, oops, go back. In some ways, these are the values that you're most interested in. This is the vector that is driving you to do this study in the first place. Q is the number of individuals in the pedigree. Okay, notice that's different than the number of individuals measured. It can be the same, maybe N and Q are the same, but they can be different. Maybe you've measured your N are individuals in generation 10, right? And you've collected phenotypic data on it, but your pedigree goes all the way back to generation 1. So Q in this case is going to be much, much bigger than N because you're going to have 9 generations of family information beyond the individuals on which you've measured the phenotype. So A is a q/1 vector of additive effects. And again, this is a random effect because what we're saying is we just reach into your population, pull out an individual for the study, but we actually don't care about that individual. It's just representative of all other individuals in that population. You'd be just as happy to throw that guy back and pull out another one. Okay. Z is another one of these design matrices, and again, this is a design matrix that relates the vector of phenotypic measures to A. And basically, Z is frequently just a large matrix filled with zeros and ones that tell you which individual a particular A goes to. Finally, we have E is an n by 1 vector of errors. So for each individual on which you've taken a phenotypic measure, there's going to be some residual error. And in a simple quantitative genetic model, this is also where all the environmental effects go to, is into this error term. Now you can set it up so it doesn't necessarily work that way, but if you have a simple model, that is how it works. Okay, and for every phenotypic measure you've taken, you're going to have an E. So it's n by 1. You get an n by 1 vector. So for every individual measured, you get this error. So what we're doing here is we're taking the variation in Y, in this vector of phenotypic measures, and we're partitioning it into fixed effects, random effects, which include the additive values, which will give you the additive variance that you're interested in, and the error term.
So could I,
Yeah.
I think I know the answer to this. So in the actual model of random effects, we're not including, for example, dominance deviation.
No, we could, but
We could, but so in the absence of doing that, or epistasis, I suppose all of those effects go right into the E term.
no. They do not. Okay, so this is a sticky wicket. If you have effects in your population that you are not modeling, and they contribute to relatives looking similar to each other, having similar values, that's going to go in. That's likely to show up in A. You're going to inflate your additive effects because you were unable to partition out the dominance variance. So that's why if you have that level of control, if you're setting up a breeding design, you want to do a half SIB or half SIB full SIB breeding design so that the additive effects do not contain the dominance effects. And the best way to get rid of that is the way that you set up the design. But if, let's say you do a full SIB analysis and you cannot partition out that the dominance effects, it ends up in A, and it's going to inflate your additive variance. And that's why if you do a full SIB, if all you can do is a full SIB design, that in the paper you write about that, you have to say, I understand that I'm not actually measuring narrow sense heritability of VA over VP. I'm measuring broad sense, which are all the genetic effects lumped together over VP.
So when you specify full SIB, when you're using full SIBs in the model, do you specify that you're producing additive effects? You don't even use that coefficient of a half that you use in this, isn't it a half?
No, you don't need to.
It doesn't go in there. So it's
No.
all of the among full SIB component of variance is attributed to A.
Yes. Yep. Right. So that's why you need to be very cognizant of what the family relationships are. Now, frequently what happens with these models, you're using this in part because you have a deep pedigree. So even if you have full SIBs in the generation in which you've measured the phenotype, if you've got, first cousins and second cousins once removed and, nephews and nieces all in your measurement population, because you've got this deep pedigree, that actually will help partition away some of that non-additive variance out of A and over and into E. So that's the advantage of having these deep pedigrees, is having those complicated family relationships can help give you a cleaner estimate of A, even when you've got a full SIB design. Yeah?
Including genetic similarity along with the pedigree help?
Genetic similarity, what do you mean by that?
I mean like marker-based similarity.
Yes. Okay, so that's a great question. So there's two things I would respond to that with. If you've gone into a natural population and you don't know what the family relationships are, you can use markers to infer the family relationships. The other thing you can do, and this is something I serve on a committee of a graduate student in the College of Agriculture at WSU, and she's been developing some methods to, even if you know the family relationships, if you've got marker similarity, can you use that to improve your estimates of the breeding values? And the answer is yes. Okay, so does somebody have a question? Okay. So this is the animal model, and as we'll see in a minute, we know everything in this equation except beta and A. The beta, which is the vector of fixed effects, and A, which is the vector of additive effects. Now we need to make some assumptions before we can solve this. We have to assume that the variance of the error terms is the matrix I, which is a diagonal matrix with ones on the diagonals and zeros on the off diagonals, times the error variance. And when you multiply the identity matrix like that, what's the so-called identity matrix times a scalar, you just get the scalar out on the other side. And what that means is there's no covariation among error terms. So the error term in individual 1 is independent of the error term in individual 2, which is independent of the error term in individual 3. Yeah?
So but that's also assuming that there's no sort of family level error, that the errors in families.
Right. You also have to assume that the covariance between A and E is 0. So what we're saying with this term and this term is that the error is independent of family effects, and it's independent among individuals. So that is an assumption that we're making here. And again, that comes in with, if you're setting up a breeding design, you want to make sure that those errors are independent. So you want to make sure that you've set up your design in such a way that you're not getting a covariance between family and something related to the error. For example, an environmental effect.
You might raise all the individuals in separate containers.
You might raise them all in separate containers, or you might, what you wouldn't want to do is have one family in this container, another family in this container, another one in this one, because then you're going to confound the container environment with family, and you're screwed. You are going to have a covariance there.
Screwed, is that a technical term?
That is a technical term. That is a technical quantitative genetics term. The other thing that we is going to assume is that the variance of the vector A, so the variance of the additive effects, is equal to this capital A times the additive variance. This capital A is the relationship matrix. It's a big matrix that tells you how all the individuals in your pedigree are related to each other. And we'll take a look at that in just a minute. Okay?
So, I will stop interjecting. But that's where our information in those, what we're calling the R coefficients.
Yes.
In my talk. That's going in, those R coefficients for the different kinds of relationship
are going into A.
And that's where you're taking account of the fact that double first cousins have less similarity and less shared genetic variance than your brother.
Exactly. So this matrix A, which is frequently also called the numerator relationship matrix, contains all that specific family relationship information. Yeah.
Just before you move on, I think I'm confused about beta. So because I have beta, like an example would be location 1 versus location 2.
Mm-hmm.
But then how do you solve for beta if that's...
Okay. Stop in the name of love. We'll be there in just a minute. Okay. All right. That'll be the next slide up. Okay. What's that? Yeah. I went back a few years before Madonna for that one. Okay. Now, here's a sticking point that seems weird when you get started with this, and it can be a pain in the butt, even once you get used to it. You also, in addition to these assumptions, which I think make some biological sense, here's a thing where you assume that you already know what the error variance and the additive variance is. You have to assume that to even be able to get started solving the model. And the reason for this is you need a starting point to begin the iterations, whether you're doing likelihood or Bayesian approaches. And so it seems counterintuitive. This is the information you want. but you're assuming you know what it is to even get the model started. And so a good rule of thumb for this is I simply always estimate the phenotypic variance, and then I cut that by half or 3/4, and I use that value for both sigma squared E and sigma squared A. That gives a starting point for the model to begin its iterative solving process, and then you'll come out the other side with something that can be very different than what you assumed at the beginning. So this is basically a statistical requirement that you assume that you know what it is, and you don't actually assume you know what it is. You just pick good starting points to get the model running. Okay, but this seems really counterintuitive. It's always so much fun when you talk into the microphone.
I don't know if I understand it correctly, but you said you use the same values for both the error variance and additive variance?
You can.
The same values?
Uh-huh.
So alpha is going to be like 1,
That would make alpha 1.
and that's okay?
Yes, that'll work, usually.
So you don't even need to estimate the phenotypic variance.
Well, except that most programs don't let you enter alpha. It asks for specific values of E and A, and if you're only working on a univariate trait, if you're just solving for one trait, you only need one value of what A and one value of E. If you're working a multivariate problem, you have to estimate not only the variances of each of the traits, but the covariances between them. And so under those circumstances, then I go ahead and estimate the phenotypic variance covariance matrix for the data set, and I use some fraction of those as the starting values for both the additive effects and the environmental effects. And generally that works pretty well. Yeah. Sure. Oh, you've got 2 microphones.
All right, so you're estimating values for sigma E squared and sigma A squared. When you run the iterations, is it moving forward by a set amount, or is it just based on the...
No, so depending on what procedure you're using, initially it may move forward. So in a likelihood situation, the first couple of iterations, it may jump a whole lot. And then as it approaches the maximum, then it's going to just move very little tiny pieces until it gets to the maximum. Other questions on this? So I'm going to show you one more slide with a bunch of this kind of stuff on it, and then we're going to do a very simple example, which I think is going to hopefully clarify this, and then we'll come back to the general situation again. All right, so your question was, how do you solve for beta and A? And luckily, a very clever guy named Henderson came up with a way to solve these mixed models. And it very neatly partitions your unknowns from your knowns. So here, these are all the terms that we just discussed. And in solving the problem, this is what it looks like. So what we've done is we've isolated the fixed effects and the random effects into now one vector, okay? So this vector now contains the regression coefficients for the fixed effects and the breeding values for each of the individuals. That's isolated from all of this, which you know. You know these design matrices, the Z's and X's over here. A lot of them are transposed. That's what that little asterisk or little apostrophe means, right? So you take if your matrix looks like this, you transpose it. The -1 for the A means you invert it, which is actually a difficult thing to do, but luckily computers can do it pretty well. So you have to invert that relationship matrix. And then over here on the other side of the equal sign, we have the design matrices times the actual phenotypic data. Okay. All right. So let's work through a very simple toy example where we only have 5 individuals and it's easy to see everything and see how this actually works. All right. So here we have a very simple data set we have 5 individuals in the data set. For some of the individuals, we know who their parents are, and some of them we don't. Every individual belongs to either herd 1 or herd 2, and every individual has a phenotype. So you could have this be whatever you want it to be. It could be, I don't know, if it's a herd of chickens or something, I don't know, herd of cattle, maybe it's the weight of the steaks or something. Who knows what it is. It can be whatever your favorite meat trait is, have that be that. Okay. So what we're saying then is, so we've got the 5 individuals. We have a system of linear equations, right, that will help us build our vectors and matrices. So individual 1 has a phenotype of 78, belongs to herd 1. It's animal number 1, right? It's the ID number 1, so it's animal 1, and it has its associated error that goes along with animal 1. Animal number 2 has a phenotype of 83. It belongs to herd 2. It's animal number 2, so that's easy, and error number 2 goes along with it, and so on and so forth for the other 3 individuals. Okay. Is everybody happy with our toy data set here? Okay, well, let's start looking at what all these vectors and matrices are then. Okay, so again, here's our toy data set. Now let's look at the things that we know. The vector of phenotypes is easy, right? We know that. Why? It's 78, 83, 70, 86, 77. It's just the list of phenotypes that you measured in the order of the IDs of the individuals in the study. So one way to think about this, if you wanted to think about this in an Excel spreadsheet, this would be phenotype, would be the column, and the rows would be individuals. Okay. All right. That's easy. Now, the next part, here's one of these design matrices, X. Now, again, the rows are individual 1, 2, 3, 4, 5. Column 1 is herd 1. Column 2 is herd 2. If it belongs to herd 1, it gets a 1, and otherwise it's a 0, and vice versa. So individual 1 is from herd 1, so it gets a 1 for being in herd 1 and a 0 for being in herd 2, because it's not in herd 2. Does that make sense? Yeah.
Are these always going to be sort of binary matrices?
Yep. Almost, yeah. So all this is doing, it's all zeros and ones because it's simply telling you what group an individual belongs to. Individual number 2 is in herd number 2, so it gets a 0 for belonging to herd 1 because it doesn't belong to herd 1. It gets a 1 for belonging to herd 2 because it does belong to herd 2. And individual 3 is the same, it's a herd 2. Individual 4 is a herd 1, and individual 5 is a herd 2. Yes?
Why do you need 2 columns to specify that? Why can't it just be like my question was, how come you need 2 columns to specify the herd if it's just 2 options? It could just be 1 or 0 in the first column and then...
Because you're going to end up with 2 regression coefficients, one for herd 1 and one for herd 2. So as we'll see, the fixed effects influence the mean value of the population, and the different fixed effects contribute a different amount to that mean. If you had 3 herds, you'd have 3 columns. If you had 10 herds, you'd have 10 columns.
Quick follow-up.
Sure.
So when you code something in R, like maybe a factor level that's A, B, or C or something like that, is the analysis reading that as kind of this sort of matrix where you have,
yeah,
if you're A, you satisfy a condition for A but not B or C?
Right. So
it's the same sort of thing.
these design matrices are not exclusive to the animal model. Any linear mixed model that you use does this, right? So, you know, we're talking about this in the context of the animal model, but this holds true for any of these models that you want to solve. Yeah. Just because animal breeders came up with it, and so they said, well, we're animal breeders, let's call it the animal model. If it had been plant breeders, it would have been, be called the plant model. But it works on any kind of organism. It's just a historical constraint, right? It's a phylogenetic constraint. Okay. Okay, now here comes the next design matrix, and remember, Z is the design matrix that relates the phenotypes that you've measured to the individuals. And this is even a simpler matrix than this one. The rows are, the columns are individual 1, 2, 3, 4, 5. The rows are individuals 1, 2, 3, 4, 5. Individual 1 belongs to individual 1, so he gets a 1, 0, and zeros everywhere else. Individual 2 belongs to individual 2, so you get zeros everywhere else. It's trivial, right? These matrices, ooh, they look scary. Ah, it's a Z matrix, but it's just a bunch of zeros and ones, right? It's very, very simple. Yes, the matrix that is relating the phenotypic measures to the individuals is always going to be a diagonal. It's actually going to be the identity matrix. It's going to be a diagonal of ones and everything else is zeros. And I encourage you, especially if you're not real familiar with linear algebra, Steve and Patrick yesterday provided some R code on how to solve matrices. Go in there and put in matrices like this and solve it, because basically it just ensures that when you do the multiplication, you get a value only for individual 1 in row 1 and not for anybody else, right? It's a very clever way to do that.
It's like a bookkeeping matrix.
It's a bookkeeping. Yeah, design matrices are generally basically bookkeeping. Yeah. Yes. Let's not. Yeah. Let's not go there. Yes, that makes it much more complicated. So that moves you out of landy land into function valued land. And although conceptually it's the same, mathematically it's a lot more difficult. Other questions on design matrices? Okay, here's our data again, and here are our unknowns. We do not know what the additive value is for individual 1, 2, 3, 4, 5. We don't know that. And we don't know what the regression coefficient for herd 1 is, and we don't know what it is for herd 2. So those are our unknowns. We're assuming, in this case, we're assuming heritability of 0.33. That was the guess for that. So that means that alpha is equal to 2.03. That's, again, you could assume that heritability was 0.5 and alpha was equal to 1 if you wanted to, or something like that. You could choose whatever you want. Finally, let's take a look at this A matrix that Steve is interested in. So we get our information to put into the A matrix from the relationship among the individuals in the pedigree. So we have individual 1 through 5 as rows, and individual 1 through 5 as columns. Individual 1 is related to individual 1 by a value of 1, right? It's genetically identical to itself. Individual 2 is not related to individual 1 at all, right? We don't know who the sire and dam of individual 1 and individual 2 is, so that's not related to individual 1, and it has a relationship to itself of 1. Individual 3 is not related to 1 or 2, but it's related to itself by a value of 1. Individual 4, his mama is individual number 1. So he has a value of 1/2 for the relationship, the degree of relationship between him and his mama. And then his daddy is individual number 2. So it gives him a 1/2 for that value as well. Individual 4 is not related to individual 3, so that's a 0, and it has a relationship to itself of 1. Yes?
What if it is, though, and you just don't know you don't have the sire and dam?
So that becomes a design problem, right? You can't fix that here. If what you're saying is I don't know what their parentage is, and I'm just going to assume they're not related. So that's an assumption being made. Or maybe those individuals were purchased from 3 different localities, and you're pretty confident that they're not related. But yes, that's something you need to make a decision on. If you don't know the parentage, are you going to assume that it's 0? You pretty much have to if you don't know the parentage. Yes?
Is it working? Oh. couldn't you like have a uniform distribution of coefficients of relationship, like, half, 1/4, and you do like a distribution of A matrices to have a distribution of additive values? So you don't know the information, but it can be
Oh, do you mean
certain types. It can be half, it can be 1/4, depending on the relationship.
or it
Couldn't
could
you
be-
do that?
You could. I don't know that anybody does do that. I mean, the other thing is it could be a half, it could be a fourth, it could be less than that. What if they're second cousins? What if they're fourth cousins? But that would take a long time to do. And so usually, and then even after you do that, then what do you do with that information? At some point, you have to make a value judgment of whether that's a 0 or not if you don't know the parentage. Yeah.
So when you have a population with genetic markers,
Uh-huh.
you are inferring the relationship from the genetic markers. You just put in those different places, different.
No. So this stays the same, right? You don't if you have markers, you actually add that as an additional random effect in the model, and then that matrix goes with that random effect. And we'll get to that in a minute. Yeah, that's a good question. So other questions about this? And then notice individual number 5 and individual number 4 have one parent in common, so their degree of relatedness is 1/4. Okay.
And again, that's the value that we call R.
Mm-hmm.
In my lecture, it's the value that Joe called F1 in his lecture. Pretty neat if you use 3 coordinates.
Right. Okay. Other questions about this? Okay. All right, so let's go back to our mixed model equations. Now you know what all these z's and x's are. They're just matrices that are mostly full of zeros and a couple of ones. You know what that is. And so you take this the equation here. These are the two unknowns. So we simply divide both sides by this. Notice this is a matrix of matrices. So we invert that and multiply it over here by this matrix of matrices over here. And so now what we've done is we've isolated our two unknowns on the left side of the equation. So we know all of this, and we don't know these things. Yeah?
Is the -1 the same as like a transpose?
No.
Okay.
It is 1 over. So this means 1 divided by this. Transpose means turn the matrix.
Turn.
Inverting it means dividing it into 1, essentially.
Okay.
But it is a matrix operation, so you can't just take
Yeah.
over each of the elements. There's a. But if you look at the tutorial, you'll see
It
R, there's a complicated way for asking to solve.
It's a difficult operation, and it's one thing that really slows down quantitative genetic software, is inverting matrices. Especially, like if you've got a ginormous pedigree, and you've got to invert this guy, and yeah, it gets messy. Okay, so if we go back to our toy example, here is the equation we want to solve. So now here, this is what the vector of unknowns look like. Here's the regression coefficient for herd 1, the regression coefficient for herd 2, and the additive effects for individuals 1 through 5. If you do this operation here but don't invert it yet, this is what your matrix looks like. Okay? And then that's going to have to be inverted. And then over here, then this is what we get by doing the multiplication, the matrix by vector multiplications in here. And notice that what happens, because remember Z is a design matrix where it's a bunch of zeros, except where you just identify individual 1 as being individual 1. So that doesn't alter the phenotypes at all. So the phenotypes actually show up as raw phenotypes right here. Okay? And then you do the inversion and the matrix to vector multiplication, and you come out with a bunch of values, okay? So you get the regression coefficients for the two herds, and you get the additive values for each of the individuals in the population. Okay. Now notice that, okay, yeah, never mind, I'll leave that alone. So, okay, any questions?
Just a comment. So if this really was state weight,
Mm-hmm.
and we see that individual 4 has the largest breeding value, then we would be possibly in a position to sell the sperm if that was the bull,
Right.
so that and people would buy that sperm because they want to produce offspring with a better than average state weight.
Right.
So that's what the cattle breeder, why the cattle breeders are interested in this output. And this output is actually in the brochures
Right.
for selling cattle sperm.
Yeah, they will give, for all these different agricultural systems, they will actually tell you what the breeding values are. And breeding values is agricultural terminology for additive genetic value.
I have a comment. Can I be heard?
Yes?
If you just use Pat will be aware of this, but if you just use the breeding value to sell the sperm, that doesn't take into account the error around that breeding value. You don't want to buy sperm that looks good but has a very high variance.
Mm-hmm.
That's
Did
a
you-
rule-
did the
that-
question-
Keep that in mind when you go to the sperm store. Other questions about this? Yeah.
Now that Joe's watching, you have to be careful.
Just a comment that I heard that breeders use some selection indexes, right?
Uh-huh.
Some indexes that put the additive genetic value, but also other things in a linear equation to rank the individual of the experimental population to
Uh-huh.
sell the sperm or whatever.
Yes, so there's all kinds of different selection indices out there that breeders will use to say, my bull is better than the one down the street, sort of thing. They, if you take all these different things into consideration. Yeah.
Are the terms, the H terms and the A terms, are they on the same scale? So can you reasonably say that herd has a much larger effect in determining stake weight than individual additive genetic
So
variance?
this, as we'll see in a minute, so these are basically defined in terms of deviations from a mean. So this is contributing to the mean, and this is contributing. Then these are going to give deviations from the mean.
Right.
Right.
Okay. So you if you wanted to maximize stake weight, just following up on Steve's example, you'd want to rear the progeny of individual 4 in the first herd environment, sort of thing.
Well, that may not work. If you have genotype by environment
Right.
interactions,
Okay,
maybe it
yeah.
does worse there.
Right.
Right. So you have to be careful about that kind of thing.
Got it.
Yeah.
So
If you start talking, it'll turn on.
It works like that?
Yeah.
Okay. So with the additive genetic variance or the breeding value, is that dependent upon the allele frequency within the population? So if you're selling sperm,
Yeah.
does there need to be some standard as far as a reference population?
I actually don't know how they do that. But one thing that we didn't have time to talk about yesterday is, and we were talking about this at breakfast this morning, the additive value contains within it the allele frequencies in the population, the P's and Q's, by definite, if you go back and derive that. And so, yes, that means that technically these estimates are population specific. Yeah.
calculation, you see that H1 and H2 is much more than A's.
So when you're selling sperm, does the A's really matter because H is going to be so high anyway?
A's are the deviation from the mean. If you wanted, like individual 4 would actually be a value of what, over 80.
Yeah, but it's 79 for everyone else. So
Right.
so they do take care of the small advantage due to the A's.
Yeah, but that goes back to a point Steve made yesterday. If you're only interested in you're interested in the variation. If there's not variation, right, then we have nothing to talk about, right? So that's one of the curious things about something might be completely genetically determined and have no variation, and so then quantitative genetics has very little to say about that, as Steve was pointing out yesterday. Yeah.
If I wanted to estimate H1, A2, ignoring all the information of the relation between the individuals, like if I just use 2 columns for my data set and estimated regression, would it be the same values?
Yeah, probably. So if you're saying you're going to not have a random effect, yeah, this would probably all end up in the error terms.
So it will be the same H1 and H2?
It would be approximately, I mean, within, with some amount of error, but yes. Because what you would be saying then is, I don't care about this, and that should end up then in that E term. Other questions? Okay, well that was a very simple example. We were only interested in one random effect, the additive effects, and only one fixed effect. If you want to add more, if you have more fixed effects, let's say you've got location and temperature and humidity or something like that you're going to treat as fixed effects, that's relatively easy to take care of. You just increase the length of that beta vector. It just gets longer and longer the more fixed effects you add. However, if you add more random effects, each one adds a new term consisting of a vector of random effects times a design matrix. So that gets back to, I believe, Roberto's question about including marker information. The example I have for you are, what if you want to look at maternal environmental effects? Maternal environmental effects is something that are going to make siblings resemble each other more strongly than non-siblings. And so, especially if you're working on mammals, maternal environmental effects are something that you probably want to model, because if you don't and they're there, they're going to inflate the additive effects. So if you add maternal effects, here we have our animal model with maternal effects. We still have Y, we still have X beta, we still have ZA, and we still have E. That's all the same as we had before. But now we have this new term, WM, where M is an F/1 vector of maternal effects, where F is the number of dams in the pedigree, and that's a random effect, and W is an N/F design matrix relating Y to M. So just as everything we talked about for the details of this random effect are going to apply to that random effect. Now we have to solve for beta and A and M, and we make the same assumptions that we did before with some additional ones. We assume that the variance in the maternal effects is, again, the identity matrix times the variance of the maternal effects. So that is, we're saying that the maternal effects are independent from each other, and we are assuming that the covariance between the additive effects and the maternal effects and the covariance between the error and the maternal effects are independent. So we're saying that A, M, and E are independent of each other. Now, I don't want to go into detail on this. I just want to put a bug in your ear. There's also something, we're saying these are maternal environmental effects. There's also something called maternal genetic effects, where the genotype of the mother is influencing the phenotype of the offspring. So some amount of the variation among offspring is not caused by their genes, but by the genes of the mother. In that circumstance, then, the variance of M is going to be A times sigma squared M. It's going to depend on the degree of relationship of individuals in the population. And under those circumstances, you usually do not want to assume that the covariation between A and M is 0. You can actually measure a covariance between the additive effects and the maternal genetic effects. So just put that in the back of your mind in case you ever need it, because you might need it someday. And again, you can. It's easy. It's easy to do. You just add in this other random effect. But now, instead of assuming independent maternal effects, you assume maternal effects that depend on the degree of relationship.
Question?
Yeah.
Or a comment? Well, it is a question. So I'm trying to remember the guy who, is it Wilburn? It's not Wilburn. This is a particular model of genetic maternal effects that you're talking about here, right?
The environmental ones or
the genetic?
The genetic ones, yeah.
Yeah, what's his name?
You know
who I mean.
Yeah, I do know who you mean.
Okay, so there's a follow. So in that model, it's sort of a peculiar thing because the trait itself is having a maternal genetic effect on itself. But the other possibility is that the female has some inheritable trait, some heritable trait, which also exerts a maternal effect on the offspring, and then the offspring pass along that trait.
Yeah,
That's
exactly.
The Landy Kirkpatrick-Landy model would be an example of that. Can you implement the Kirkpatrick-Landy model of genetic maternal effects?
Yes.
You can?
Yeah. So all you do under those circumstances, instead of the variance of M being the identity matrix times sigma squared M, it becomes A times sigma squared M. So in other words, if you have that kind of maternal genetic effect, two mothers who are sisters are going to cause the effect that they have on their offspring through that maternal genetic effect is going to be more similar to each other than two mothers who are not related to each other.
Right. So you make this assumption about the inheritance of the traits being wholly additive, basically.
Yes.
In other words, there's a resemblance. So you're throwing away some of the. What are you doing? What are you doing to the dominance and epistatic contributions to? We're getting into an arcane world, but if you get into maternal effects, you've got to confront this, don't you?
If you can, it's actually so. With every additional random effect that you add, it becomes more difficult to solve these equations, and you need a bigger and bigger sample size. And so, generally speaking, what ends up happening is you run out of power to be able to add these sorts of terms. So maybe you can get in a maternal genetic effect, but once you've done that, you probably don't have room. You don't have the power to add anything else in there and be able to adequately solve the problem, the equation.
If, for example, you imagine that all possible traits exert maternal genetic effects on offspring, that's a huge it's a huge specification, isn't
right. I mean, you need to have like 1 million offspring that you measured or something like that. Yeah. And then I'll just show you, we don't need to belabor this, but if you've added an additional random effect like this maternal effect, the basic look of the Henderson equation is the same. You just now have this extra term in here and extra terms over there. So it's all solvable. So with basically with every additional random effect that you add, you're adding another layer of complexity to solving this problem that's going to eat up degrees of freedom and that sort of thing. So that's why you start to run out of power if you add too many random effects. And that's the reason why if you've measured gender in your population, for example, and you're debating whether to include it as a fixed effect or a random effect, and you could go either way, a fixed effect is going to be computationally a lot more simple than adding it as another random effect. Okay, how are we doing on time here?
I was just looking at that. We've got time for, are you done?
No.
Your discussion for this lecture goes to 10 till 10:15.
Okay. We've been discussing as we've been going along, so hopefully I'll just... Okay. So the next thing I wanted to do, I want to go over this just real briefly because you're going to get a whole lecture on it tomorrow. Is how do you solve these equations? And one way is using maximum likelihood, which will be talked about tomorrow by Brian. And with likelihood, you have the conditional probability of the data given the parameters mu and sigma squared. Mu is the mean. It's just x beta. These are the fixed effects. And then sigma squared is going to be this term here. You know what all those things are. That's the genetic variance plus the error variance. Okay? So the likelihood is the conditional probability of the data given these parameters. And remember we talked about the starting values. So what happens with the likelihood approach, you've selected starting values for your sigma squared E, sigma squared A. It goes through, does the likelihood maximization, and it gives you then your values of A and also beta. I left that out, which gives you then a new estimate of sigma squared E over sigma squared A. Now we've moved away from the starting values. Then you take that, and those becomes your starting values for the next iteration, and so on and so forth. And you just rinse and repeat until the likelihood is maximized and the parameters stabilize. Okay.
Question?
Uh-oh.
We've discussed this before, Pat, but and I'm glad you got this presentation of likelihood and Bayesian. The parameters in the likelihood inference here are the sigma squared A's and so on. But the breeding values technically aren't parameters, are they?
No, they're not. So that's why I put this up here. So the parameter mu is just x beta, the design matrix times the regression coefficients. And the sigma squared is the design matrix Z times the numerator relationship matrix sigma squared A, Z transpose, plus the identity matrix times sigma squared E. So those are the parameters, right? The individual things are not, the individual A's are not the parameters that are being used.
So they're sort of expected values conditional on the current values of the parameters, which are
Yes.
when you're doing likelihood inference, it's the parameters that officially you're searching for parameters, although here your interest is also in individual breeding values.
Exactly.
Okay.
Just a follow-up question.
So sigma squared sub A, is that the additive genetic variance
Yes.
estimated from the output breeding values, A values? You see what I'm saying?
Yes, yes, it is. Yes, that is what's going on. But this is the parameter. This, I think of these as kind of maybe being the side product of that.
Yeah. So you're holding. So you hold that sigma squared sub A constant, right? That's something you solve for, and you launch into likelihood to solve for the optimum set of breeding values under that.
Well, no. What you're doing is with each iteration
I
of
see. There, I missed
you're
it
getting
there.
new
Yeah,
ones.
yeah.
You're getting
Okay,
new ones.
I see it. Okay, yeah.
So
right.
with each, so you start,
Yeah.
like with the program, you start it running, and you can watch it do its iteration. With each iteration, you get a new sigma squared E and a new sigma
okay.
squared A,
I'm sorry.
and then it does it again and again, and it's running up the likelihood slope.
Yeah, it's right there. I just didn't see it.
Okay, so that's the likelihood approach. The Bayesian approaches are frequently used, and again, Brian's going to talk about this extensively tomorrow. And with Bayesian, you're talking about the conditional probability of the parameters given the data. So kind of that intuitively makes more sense than the conditional probability of the data of the data given the parameters. So that's a little bit more intuitive, except that, whoops, damn it, come back here. except that this is proportional to the likelihood, which we just had, times the the prior belief in possible parameter values. So we're kind of back to the same conceptual situation that we had before in this sense. But what solving this does is this produces what's known as a posterior distribution, which is a probabilistic distribution associating each value of a parameter to a probability. And you need an iterative process to solve this. And the one that is most frequently used is, and certainly in R for the module we're going to use, is MCMC, which is Markov chain Monte Carlo. And I'm sure Joe could talk to you about that for a long time, and I'm not barely going to talk to you about it at all. So this is an algorithm based on the proposal of a new value for a parameter as a function of the value of the other parameters at each iterative step. So again, this is going to be a lot of iterations. And then you save the value of the parameter at each iteration, getting a series of values, which is the posterior distribution of interest. So you get a posterior probability distribution for each parameter. Okay. And we'll be looking at these in a few minutes once we start the computer exercise. Okay. Just a couple of other things. So remember how one of the assumptions that was made yesterday and that we've been making so far today is that the traits that were to which we're applying quantitative genetic analyses need to be Gaussian. But what if your trait of interest is non-Gaussian? What if you're looking at, this is from a population of wrens, what if you're looking at the total number of offspring produced? That's definitely a non-Gaussian. Can we estimate variance components? And the answer is we have a good shot at doing that now using what are known as generalized linear mixed models, or GLMM. And this is a combination of generalized linear models and linear mixed models. And these should be able to handle non-Gaussian traits. It requires the use of a link function in order to do this, but otherwise it's still using an animal model, and this definitely requires Bayesian approaches. So what we're going to see with MCMC GLMM in R, we're not going to do it today. We will be using Gaussian traits for our class exercise, but you can actually use those on non-Gaussian traits. And I've tried this with varying levels of success, and there's kind of some weird assumptions you have to make, but you can at least give it a shot. So this is Dunatar Castle in Scotland, sitting right there on the North Sea. And my colleague in Scotland refers to MCMC GLMM as Mick-Mick-Glim, which I always kind of like that. So with Mick-Mick-Glim, you can estimate variance components for any Gaussian or non-Gaussian trait, at least theoretically. Again, it's not clear quite how well that works. You can have any number of fixed or random effects. You can handle complex pedigrees, and it's very flexible. The downsides to Mick-Mick-Glim is you need priors, and the tradition, or what most quantitative geneticists do, is choose uninformative priors because they don't actually have any prior information about what the variance components are in that population that they're studying. So usually uninformative priors are chosen, but there's a lot of debate about what's the best kind of uninformative prior to use. And I don't want to go down that path today, but I gave you a couple of papers to look at, and some of those begin to address this issue of what's the best kind of uninformative prior to use.
Does this sort of fall into the category of garbage in, garbage out? Like if you have a bad prior and you don't know about how bad it is your
output bad? So that's something you can play with. So for example, if you and this is actually for evolutionary biologists, this is a big problem because we usually have relatively small data sets, which means the priors can have a huge effect. So if you take a data set, and actually I think in one of the papers they give you, they sort of point this out. If you have a data set that's relatively small, put in an uninformative prior and look at the output, and then put in like a strong Gaussian prior, and you'll get completely different answer. So yeah, you can really harm yourself with the priors if you're not careful. Yeah.
The one prior to be very wary of is a flat prior going out from 0 towards infinity, because technically speaking, it's not even a distribution at all, but it tends to, by allowing values that are very, very big, it tends to basically overwhelm your data.
Mm-hmm. So as we'll see, McGlim uses what's called an inverse Wishart distribution for setting priors. That's its default mode, and we'll look at that in a few minutes. Yeah.
If you take a Bayesian approach like this and you have to specify priors for your parameter values, is this a way to like bound your heritability estimate, for example, between 0 and 1? Like yesterday we had
a
lot of concern about getting those impossible parameter values, so this doesn't really help you there?
No, this is not going to help with that. And
Okay.
You know what? I'm not concerned with that, with getting values that are less than 0 or greater than 1, because if I get that, then I know that probably I don't have sufficient power to solve the problem. It's like that's an answer. That's a really strong answer. If I get a heritability of 1.5 with a plus or minus 2 or something, it's like, all right, I can't get heritability in this with this data set.
So there are some, your question actually comes in the likelihood framework usually, because the likelihood of getting a negative number in a lot of these formulations is 0. And so what will tend to happen is in some likelihood analyses, then any estimate that's less than 0 will just be set to 0, because the prior likelihood is 0 that it can actually, that a variance can be negative. And so I'm not an expert in this, but it creates sort of this discontinuity around 0. And so frequently when you see printouts from a likelihood analysis of variance component estimates, you'll get a bunch of zeros, whereas if you did it using a general linear model sort of framework, you'll get these negative estimates. And that's simply because they're just set to 0 if they're negative, because you know that they can't be.
Yeah.
I don't know if that creates bias or not.
Yeah.
Can you treat the priors in a Bayesian fashion and try and look at the posterior estimates as a function of different
Yes.
priors?
Oh, absolutely. And that's not a bad idea. You know, especially when you're. I mean, it takes. Getting variance component estimates takes a while, and you need to just like relax and say, all right, this is going to take me a month or whatever. And as you get started, if you're using Bayesian approaches, yeah, you might want to use different priors and see how using the different kinds of priors influences your posterior distribution. Are there certain priors, as Joe was pointing out, that are going to overwhelm your data? And you should look for those. Find one. Wipe out your data with your priors to see what it looks like, and then try to find better ones. So there's kind of a lot, kind of what you spend 90% of your time like fiddling around and getting the model exactly how you want it, including the priors, and the last 10% of your time actually running the model and getting the information you need. So you want to investigate all these things. Yeah.
So that's a good piece of advice, to stay relaxed.
Yes.
Stay calm.
What's that? Keep calm.
I know Yanis Antonovich calls this genetics without tears. You start with a simple, you just stay relaxed, and you get negative estimates. It's not the end of the world. You don't cry about it.
Gin helps.
This is Steve's life philosophy in general. Can I make a brief philosophical statement that has nothing, not the Bayesian versus whatever, but actually about statistics, and that you guys in your career, and you're sitting here, it's like, this is overwhelming. But it does show you, everything that Pat's been saying shows you the importance of actually understanding what you're doing as you go along. So when he says, oh, try this prior or that prior, and then figure out which one makes sense, and that takes a lot of you driving the car at that point. And so what tends to happen when students first start off is the statistics drives them, and they know that they have to do this, and then you get this p-value, and then you interpret your data that way. And what you want to progress through in your career is getting to a point where you feel enough ownership that you are driving the statistics, so it's doing what you want to do, and if you get a result that doesn't make sense to you, then you figure out why, or then you can say, oh, I wanted to try this prior or that prior, and then you have some sense of that it's not just complete magic, even though a lot of it is going on in a magical way. And I think that that's one of the hard career transitions that people have, is to get that sense of confidence about this. And it's actually not that common. And that's part of the strength of quantitative genetics, is that you can't make it further here until you sort of push that barrier and then try and move beyond it a little bit.
Yeah, good point.
I think in a Bayesian analysis, if you put in a prior on your variance components such that they're never negative, you can't come up with a negative heritability, right?
I think that's true because so what I don't use MC GLMM all that frequently. But when I have used it, I've noticed that I've never gotten negative variance components. They basically get very, very, very close to zero, and then they'll have gigantic errors around them. And that, to me, that's the indication that if it could have done, it would have given it a negative value. But yeah, I have not seen negative variance component values from McMick-Glim. Okay, so we're almost at the end of the discussion time. We'll take a little bit of a break in just a second, and then we'll get into using McMick-Glim. Before we do that, I just wanted to tell you about the data that we're going to be looking at. I've done some consulting work for the largest rainbow trout farm in North America. This is Clear Spring Foods. This is just one of their rainbow trout farms. Here's a tractor trailer. And look how big these raceways are where they raise trout. They have their own processing facility. They can convert 60 tons of live rainbow trout into fillets in one day. It's amazing. And so, whoops, I'm going to zip through this real quick. What they're interested in is they want to have rainbow trout that are more football shaped because they go through the slicer machine better. Okay, they go through the filleting machine. Again, these are farm trout. These are not hatchery trout or what, these don't live in streams. They live their whole life in these concrete tanks. So they want fish that are more football shaped. And so we collected a bunch of data. We collected data on about 1,000 fish, took photographs. We did some initial principal component analyses. And the method we came up with is we just took some of the simple body size measures that we had: head length, body depth, and body mass. I wanted to get estimates of the additive variances and covariances for these traits, and then predict the multivariate response to selection on increased body depth, because they want to have a deeper fish. So the data set that you're going to be using has 900 and some fish, or 860 fish with a moderately deep pedigree. I think it goes back about 3 generations, and it has all these data in it. We're going to start off just doing univariate analyses, and then we'll move into multivariate analyses, and you can see how GLMM is set up. So you'll be using a real data set here. GLMM takes a long time to run to get good estimates. We don't have 12 hours to sit here and do it. So we're going to do very short runs, and we're going to have very sloppy looking estimates. But when you go home, you can, as you use it on your real data, you can set the number of iterations to be high enough that you get more robust estimates. So let's take a 5-minute break to go to the bathroom, get a drink or something like that, and then we'll walk through that R code that I posted on the course website, and we'll see how GLMM works.